everyone. Thank you for the opportunity to present to you today. My name is Ryan Carlo. I'm the program lead with the Greenbelt Foundation, and I'm joined today by a research soil scientist at the Soil Health Institute, Dr. Jenny Bauer, and Alloy Thompson, certified crop advisor with the foundation. We're here to present key findings on measuring soil health in Ontario from our soil health program. An outline for today's presentation. Our primary goal today is to transfer program learnings to help inform the work that you all do in soil health. And we'll be doing this by telling you about building our program farmer reports, detailing the reception and feedback we've been hearing during one-on-one farmer meetings, and then finish by assessing the soil health frameworks that we used in our program. So a bit of background on the work that we've been doing. In 2023, the foundation launched this 4-year pilot program to provide soil health assessments to farmers in the Greater Golden Horseshoe region of Ontario. This program involved a key partnership with the Soil Health Institute to bring their scientific expertise to the region. And the program has an advisory committee of key agricultural organizations who advise on implementing the program, including representatives from OMAFA, whose partnership and advice has helped direct our program and transfers learning to the province's own soil health work. There are several goals with this program, including piloting 2 soil health assessment protocols, SHAP from OMAFA and the SHI's Essential Suite of Indicators. Educating producers and advisors on soil health testing is also another goal. And finally, increasing the use of BMPs across the Greater Golden Horseshoe region. This is being done through 2 primary methods. The first is the creation of a detailed results report given to our farmer participants that includes a soil health management plan, and we're also providing support through our certified crop advisor. Finally, this program aims to contribute to the Ontario Soil Health Database and future soil health testing work in the province through activities like today's presentation. So for participants in our program, we targeted our recruitment looking at grain and oilseed farmers across the Greater Golden Horseshoe region. We focused on those growing some type of corn or soybean in rotation, as that's the most common type of crops grown on the most acres in the region. Those who signed up received a soil health report with a data privacy guarantee. And farmers participated for a number of different reasons. Some were looking for baseline information, some were trying to compare fields, those they had worked for a number of years and those that they had newly rented, and they were unsure of the management history. And some were looking for proof that over the years, the practices they have implemented have resulted in increased soil health. Many participants were also looking for opportunities to connect with other farmers around the topics of soil health testing and BMP use. And with that, I'm going to pass it to Jenny to talk about the methods that we used in our program.
Thank you, Ryan. So before I give an overview of our study and the results, I want to talk about why we measure soil health. So soil serves multiple functions on the farm. It helps plants grow, cycles nutrients, stores carbon, allows water to enter, supports plants and livestock, resists erosion, drains well, stores water, and resists pests, pathogens, and disease. So the goal of a soil health test is really to measure and improve the soil functioning. And as my boss recently coined, and you got to measure what you treasure, I think. So that's part of why we implemented the soil health testing. We sought to benchmark 1.8 million acres of grains and oilseeds around the Greater Golden Horseshoe. And we selected sites to sample by grouping soil by the particle size class of the uppermost soil, by the drainage class as mapped, and by management. And to do that, we collected an initial management interview and then later 5 years of detailed management history. And the intent with this was to select common row crop soil so that our results could be meaningful to farmers who were on the dominantly farmed soils of this region. We ended up including coarse loamy, fine silty, and clayey soils. So really, there's just a broad textural gradient across this region, and a little bit of a difference in climate as well, but we were targeting well-drained and intermediately drained common particle size classes. We ended up sampling 285 sites in 2023 and 2024, and the concentration is shown by colors in the in the map of sample locations. And I've also showed a texture triangle of all of our sites here, showing that texture gradient that I just described. So a lot of textural diversity, but luckily it represented a gradient that we then were able to use when analyzing the data. And I mentioned we looked at different managements. So initially we took a rough pass sorting row cropped soils into baseline sites, soil health practices, and reference sites. So these baseline sites usually implemented annual tillage without cover crops. And they looked like the video you just saw with little residue. Whereas we also sampled soil health practices, which use no-till or minimum till with winter wheat or cover crops, lots of residue. You can see some green cover crops here and standing biomass. And then to have a maximum estimate of soil health, we also sampled reference sites. These included long-term perennial pastures and fence rows, as well as even some orchards and woodlots. But the goal of this was really to capture the maximum soil functioning for the soil types that we sampled. So Ryan mentioned that we included 2 soil health assessment frameworks in this study. And as part of that, we measured a bunch of soil health indicators. These included the SHI, Essential Soil Health Indicators, as well as the SHAP Analytical Indicators. And the SHI indicators included organic carbon, aggregate stability by image analysis or SLAKES, 24-hour respiration, and available water holding capacity. However, we substituted 24-hour respiration with 96-hour respiration to match SHAP, and available water holding capacity was actually estimated using a function based on organic carbon and texture. SHAP included organic matter, but we ended up calculating that from organic carbon by 1.9. We also measured active carbon, aggregate stability by wet sieving or Yoder, 96-hour respiration, and potentially mineralizable nitrogen. I'm not going to go through all of these because it would take a while, and I'm not even going to show individual results of all these, but it is an awesome data set. So that brings me to the role of the database and our data management plan. So this database includes indicators from both frameworks as well as soil properties, including measured soil texture and pH, and a detailed 5-year management history for each sample. And that management history enables calculation of different tillage or living roots indices, but also gives flexibility in analyzing. We did constrain our sampling to the dominant soils planted with grains and oil seeds. And this database is available for sharing with partners, including OMAFA and universities, but we have a data sharing plan that governs sort of the guidelines around that. And that's in our data management plan, which govern data privacy and again, considerations around sharing the database. But our goal was to really maintain confidentiality for producers and comply with PIPEDA and other regulations. So with that, I will turn it back over to you, Ryan. Thanks.
Great. Thank you so much, Jenny. So following all the data analysis work, we provided farmers who participated in our program with a comprehensive soil health report detailing the sample results, soil health indicator definitions, as well as information on the sampling protocol and the data pool for the 2 different soil health assessments that were used. On your screen now, you'll see page 2 of our soil health report, which shows the SHI results presented with bar graphs representing the benchmarking approach, with individual site sample compared to the 3 different management systems on a similar soil type. And on page 3 of the report, this shows the SHAP results presented with the scoring approach for the 5 indicators in the SHAP protocol. So both of these approaches employ a much more visual way to present soil test results to farmers compared to what they're used to seeing through a typical soil fertility test. So with this information and in this report, we can give farmers more context to help them better interpret what their results mean. To accompany the results pages in our farmer report, we also developed a management plan from existing resources and guidance that was provided in the SHAP protocol. This plan is our answer to the so what question that farmers will ask once they see their individual results. As in, so what do these results mean for my farm and what practices I use? In the plan, we're turning soil health results into recommendations on how farmers can adjust practices to improve their soil health scores. The plans are tailored to individual farm operations, but do include common practices such as reducing tillage and adding cover crops into the rotation, with a focus on getting started and incremental progress rather than drastic changes. The management plan also provides links to cost share programs for BMP implementation relevant to the recommendations, as we know that cost is an important barrier to try and overcome. Additionally, this is a living document and can be updated during yearly visits with our program CCA to reflect the changes on farms or capture shifting soil health goals for individual producers. An important factor in soil health testing is the cost. The SHAP and SHI tests cost between $100 and $150 each, which is certainly higher than a basic fertility package that farmers are kind of used to paying for. The full cost of the process can be higher considering data analysis, report generation, and report delivery. Those are all additional expenses that can be sped up or reduced with automation, but still need to be considered. Additional considerations is the laboratory capacity needed to make sure these soil health tests are available to farmers. We ran all of our samples at the AFL lab in Guelph, but for wider adoption across Ontario, more labs would need to be able to run these soil health tests. In terms of costs for the farmers, cost sharing for soil health testing is used in the US and in PEI, making it a viable approach to get more soil health testing done. And now I'm going to pass it to my colleague Aloy for some information on what our farmers are telling us.
Thanks very much, Ryan. Farmers express strong appreciation and interest in soil health testing and emphasize its clear relevance to real farm operations and an interest in retesting to track changes over time. As one farmer noted, it's a good idea. When you spend money and time, you want the soil to give you a good crop. This sentiment reflects the practical value they see in the program, particularly in providing insights into the soil condition and helping them better understand what is happening beneath the surface. At the same time, some farmers observe that the interval between soil sampling, receipt of the initial report, and the follow-up meeting felt lengthy compared to the typical fertility test approach, an outcome of limited lab capacity and the nature of a pilot project. These 2 important focus points ourselves and partners to consider when thinking about scaling this work. Farmers are interested in testing, so we need timely turnaround, which requires automating aspects of the report development and building lab capacity. Next slide, thanks. Almost all farmers express a strong interest in improving or maintaining soil health, and many producers indicated an interest in future testing in order to track the long-term impacts of practice change. The soil health goal of our producers commonly center on increasing organic matter, enhancing water retention, strengthening soil resilience, and sustaining long-term productivity. Many also conveyed a deep sense of stewardship, emphasizing the importance of leaving their land in better condition for their children and grandchildren. When certain practices are not adopted, this is generally not due to a lack of motivation. Rather, it is typically the result of financial limitation, equipment availability, or broader operational constraints. Farmers value receiving soil health reports and gaining clearer insights into what is happening in their soils. For some, the findings serve as validation of the management practices they have implemented for decades, such as cover cropping, reduced tillage, and the use of organic amendments. And for others, it has helped them make decisions about their management practices going forward. At the same time, some farmers felt discouraged about receiving poor results from the assessment, especially if they had just recently adopted practices that take time to be reflected in the results, and others raised concern regarding the sampling process, particularly whether individual sampling points accurately represent condition across the entire field. This required clarification and further discussion to address their questions and ensure confidence in the results. These receptions show the importance of discussing the results with farmers and providing encouragement and guidance along with the report. What are the results of this on the landscape? 55% of participating farmers have adopted at least one new BMP for a total of 24,306 acres. A substantial portion of adopted BMPs was reduced tillage. Cover crops are also widely utilized, although the scale of adoption varies depending on weather windows, input costs, and access to appropriate equipment. In general, farmers tend to integrate these practices incrementally. Despite strong progress, several persistent barriers remain, and these include high cost of equipment, weather-related timing challenges, constraints associated with rented land, and reliance on custom operators. With this, I now turn it over back to Ryan.
I'll actually take it from here. Thank you, Eloy.
Thanks, Jenny. Sorry about that.
Oh, no, you're good. So with multiple years of sampling under our belt, we were able to analyze the data, and I'm really glad that we were able to synthesize our results, deliver meaningful reports to farmers, and then also create a regional soil health report. So this can be found on the Greenbelt's website, and basically we found that farmers who are implementing some of the practices that Eloy described are improving soil health in the Greater Golden Horseshoe. So we found that farmers who implemented reduced tillage and cover crops had on average 12% increase in water holding capacity, 28% higher soil respiration, 39% higher potentially mineralizable nitrogen, and a 16% increase in aggregate stability. And these results are currently under peer review with Geoderma, and we hope that they will be published soon. On the right, you can see the regional report, so I encourage you to go visit it. I already introduced the different indicators that we measured, but I want to kind of move forward by considering a real central goal of this study, besides just looking for improvements in soil health, we also had the unique opportunity to compare two different soil health assessment frameworks. So these frameworks govern how measurements and indicators are interpreted. There are a lot of studies that compare indicators, but these assessment frameworks are actually influenced by the underlying database, how data are grouped, and assumptions. So a few studies are actually able to compare soil health assessment frameworks in terms of how indicators are scored and interpreted by producers. So it's important to understand all the factors that might influence assessment for programming, reporting, and potentially improving SHAP and other assessment frameworks. So our guiding question was how different soil health assessment frameworks shape soil health interpretation and outcomes. So I'll start by discussing results of comparing the indicators. Then I'll move to comparing outcomes and sampling design, and finally I'll discuss the role of texture. And I should say, obviously, I work for the Soil Health Institute, but we've been collaborating with folks at OMAFA and the University of Guelph, and our goal is to do a fair comparison between approaches. So this work has been guided by a lot of thinking and analysis. First, we looked at relationships between the indicators, and we ultimately found that the carbon and nitrogen indicators were all highly correlated, and this aligns with other findings from continental scale work looking at soil health indicators on experimental agricultural sites. We found that organic carbon and active carbon had the strongest correlation of 0.7, with 1 being perfect correlation, followed by organic carbon and respiration, and then followed by respiration and PMN, and organic carbon and PMN. So we did find that aggregate stability indicators were the least correlated with other soil health indicators, suggesting that they might represent distinct elements of soil functioning. We also looked at the sensitivity of each indicator to management. So we just looked at the row cropped soils for this and the influence of inherent soil properties such as clay, temperature and precipitation, pH. We also looked at landscape position derived from LIDAR and a few other variables. We included management as our categories, so baseline versus soil health. But during our analysis, we also found that organic amendments influenced our carbon and nitrogen indicators, so we included that as a term. Respiration was found to be the most responsive to management based on this analysis. And this was using a partial eta squared analysis. I'm not going to go into the details, but you can basically interpret the numbers as an R squared, explaining the amount of variation. And the higher the number, which here are decimals, the stronger the sensitivity to management and other factors. So respiration was the most sensitive, followed by SLAKES, followed by PMN, then organic carbon and active carbon. We did find that one indicator was not sensitive to management, and that was the aggregate stability by Yoder within this study, and based on the lab we used. Next slide. So as mentioned, carbon and nitrogen indicators may capture related aspects of soil functioning and management because of their high correlation. I should say that this study used a space for time study design. And so it was not implementing repeated sampling at same sites. So tracking progress over time might yield different insights around some of these indicators. Like, is one of these indicators an early response, show an early response versus the other carbon and nitrogen indicators? However, based on our results, we did find strong correlations. In terms of aggregate stability, SLAKES was more responsive to management than Yoder, and this translates to outcomes. So I'm showing two plots on the right. So this is the distribution of SHI and SHAP scores once we sort of rate the raw values. And you can see there's pretty much separation between the brown and blue for the aggregate stability by SLAKES, whereas the Yoder tended to overlap in terms of those managements. And that led us to believe that SLAKES is less influenced by some of the, first is less influenced by manure than the carbon and nitrogen indicators, and likely captures different soil functions, but also responds to management over Yoder. Before I move on to showing the results of comparing outcomes and sampling design, I do want to mention some key differences between the two frameworks we included. So the SHAP framework is based on distribution-based scoring, similar to the Cornell method of assessing soil health, whereas the SHI method uses a reference-based benchmarking approach. As I mentioned, looking at the maximum soil health attainable for soil types by comparing to perennial long-term undisturbed soils. So the purpose of these is rather different. In the distribution-based scoring, you could say the purpose is to evaluate soil health in relation to its distribution across agricultural land uses. And the SHI approach sought to compare management-driven progress within a single cropping system, in this case, compared to the maximum soil health potential. And that led to different goals of sampling. So with distribution-based scoring, the goal is to capture the range of soil health across dominant soils and agricultural land uses, or all soils and agricultural land uses. Whereas the SHI approach, the goal was to sample evenly across the most common soils under grain and oilseeds, and the references on comparable soils. Management was treated a little bit differently in both. It was implicit within the scoring approach. Management is represented through its distribution in the data set. And in the SHI approach, it was treated categorically. And we implemented or sought to have a balanced management sampling design. And then finally, the soil properties were treated differently in both approaches. So they were addressed via texture groups in the scoring and modeled using continuous properties in reference in the SHI approach. Okay, my plot may not have shown up yet. Do you mind clicking it? Thank you. Where's my data? So now I want to move on to discussing outcomes and potential interpretation. So I should say that overall, there was alignment in terms of the ranking of the two approaches in terms of scores. We couldn't directly compare numbers because of differences in how those scores are created. But in general, high scores were high in both approaches and low in both approaches. However, SHAP score ratings did tend to be lower than the SHI ratings. And part of that goes back to some of the differences in approach that I was discussing. So, for example, there were no SHAP scores be outside of low that were also rated as low by SHI. However, some of the SHAP scores of low were rated as medium or high, even high, by the SHI approach. And this propagated through where there were some SHI medium rated soils there were SHAP scores that scored as medium that were rated as high by SHI. And basically, this goes back to how samples were evaluated. So in SHAP, they're evaluated against all agricultural land uses. This SHI approach sought to rate against other grain and oilseed soils. And then I just want to make a note about their rankings. So the black line indicates perfect relationship between, or perfect ordering of scores between the SHI and SHAP approaches. So everything would be lined up on the black line if they were the same in terms of ranks. We did find that sometimes in the case of active carbon, you can see more blue dots on the right side and more brown dots on the left side. This basically means that in general, the SHI scoring rated soil health sites a bit higher than the SHAP scores, and SHAP scores rated the baseline sites a bit higher than the SHI approach. Next slide. So as I mentioned, the SHAP ratings are probably lower for row crop soils because they're comparing to the full distribution of agricultural land uses. And the goal of the SHI approach was to kind of amplify management effects, and that was our study design. So it's not a total surprise that that those scores were a bit different in that regard. We did also find that organic amendments influenced scores for the carbon and nitrogen indicators. And when thinking about outcomes, this was interesting to consider because organic amendments may not always be strictly manageable by producers. What I mean by that is application of organic amendments may be limited by access and also distance from the home farm. So they may not be transported long distances because it's not feasible for a farmer. All right, now I want to get into some of the sampling design effects if you're not tired of seeing graphs already. So we used real and simulated data sets to construct scoring curves for medium textured soils. And the goal of this was really to look at how the composition of the database influenced how, for example, a SHAP scoring curve would look. Now I want to back up and say that the SHAP scoring curves are based on the Ontario Topsoil Sampling Program, which used a really detailed sampling plan, and I think did a really good job of representing soil and cropping system diversity on the landscape. So we simulated a data set with 20% row crop versus 80% sort of pastures and perennial sites. We simulated around 57 or 60% row crops, 75% row crops, which was the topsoil sampling program, and then we simulated 80% row crops. And I should have said the 60% one is actually based on our whole data set. So that was the actual composition. But basically the takeaway here is that scoring curves shift left with increasing composition of row crops in the database. And this tends to raise indicator scores. So if you start with 20% row crop in your database, you're going to have a score of low for a respiration value of 20. That would raise to medium or around the 50s if you have 60% row crops. And then if you boost that to 80%, you'll end up with a score of high. And we know that this is the way distribution-based scoring works, but it just, I think, reinforces that it's important to consider the sampling design when constructing these scoring functions. Next slide. We also found effects of sampling design influenced the reference-based approach. So remember that the references are used to estimate maximum soil health potential. And we found that if you split up references into managed and unmanaged soils, so considering managed as orchards or hayfields or pastures, and unmanaged references as woodlots or fence rows, we found significant differences for organic carbon, and respiration. Aggregate stability did not show a significant difference. And where this comes to bear is on, I would say, landscape scale accounting of soil health. So you might have different estimated potential soil health based on whether you're considering the ceiling to be based on these managed versus unmanaged references. We also examined the role of texture in both approaches. So first, we looked at these texture groupings, which are used to construct SHAP scores and group them into coarse, medium, and fine. And we found that there can be up to 30-point score differences at these texture boundaries, which just means that if you're a producer near those boundaries, your score could change by up to 30 points depending on if you're classified as coarse or medium at the two, or medium or fine at the 2 stars. However, this is only pertinent when comparing a soil to similar soils under different management, or if you're right at that line and you're taking and it could be due to laboratory variance or difference in hand texturing. But it shouldn't have a huge effect if you're trying to track progress over time. So sampling the same soil multiple years in a row, it is also unlikely to affect whole database analysis because continuous texture can be used, and also it's not as directly related to the comparability. I should say increasing texture groups is an option, but it does reduce the statistical power. And this was found by Inderjot Chahal in a paper looking at the effect of texture on scoring functions. And Dan Surratt has also looked into this. There's additional work by Cash as well that has done a good job exploring next steps. We also looked at the role of texture in the reference-based benchmarking approach. So remember that we used the mapped particle size class and drainage to stratify soils. However, we know that mapped particle size class can differ from laboratory analyzed particle size class. And we did find that laboratory determined particle size class was different for over 50% of samples, so over half. And that was a lot. However, we found that there were similar ordings of particle size class frequency. And what I mean by that is our dominant soils were still coarse loamy and fine loamy, followed by clayey. However, we did find that there were more fine silty soils and fewer sandy soils than mapped. Part of that was the good job at hand texturing and training that we had from our soil samplers, and including Ryan. After grouping these soils, we did use texture as a continuous variable. However, this did add costs to the soil test, as laboratory measured texture is currently does come with a high cost from the lab we were using. So I want to offer some potential learnings that could be used in improving SHAP and then and other soil health assessment approaches. First, when looking at the soil health indicators, one possibility is to consider reducing redundancy, which I realized was a very redundant sentence after writing it, among the carbon and nitrogen indicators while preserving functional coverage. Another option is to reassess the laboratory method used for aggregate stability based on demonstrated management sensitivity. So we found that Yoder was less influenced by management. There are other great aggregate stability methods out there, with the rainfall simulator being the gold standard. We did find that SLAKES was responsive to management, so that could also be an option. In terms of the scoring methodology, we suggest exploring continuous treatment of soil properties and management where possible. So the SHAP approach used texture categories and the SHI approach used management categories. But if we can use our data to model the influence of management and soil properties continuously, that would minimize boundary artifacts and also reduce some of the subjectivity in assigning management categories. Next, there could be different strategies to reduce score discontinuities if the texture categories are retained without sacrificing statistical power. More explicit treatment of management effects within cropping systems could help contextualize scores for producers. So what I mean by that is giving score ranges for grain and oilseed producers, for example, so they know what they're likely to achieve if they implement beneficial management practices. And then finally, with interpretation and communication, we suggest framing attainable score ranges, as I mentioned, for cropping systems, and potentially separating the communication of progress from maximum potential. And finally, trying to contextualize the effects of non-manageable factors, such as access to organic amendments when reporting scores. And what I mean by that is, if it's possible to show room for improvement under both manured and non-manured systems, that might be more actionable for producers who may have varying access to organic amendments. Although we know that organic amendments definitely increase soil health, it just comes with added context. So with that, thank you for listening, and we are hoping to publish this comparison in the future, but it may take a while. And in the meantime, please don't hesitate to reach out to myself or Greenbelt with questions about what I presented. I'll turn it back to you, Ryan.
Awesome. Thanks so much, Jenny, and thank you everyone for attending today. We have some CEU credits available if anyone is a CCA and is needing those. Additionally, we're going to follow up this webinar next week with a recording of today's presentation, as well as a report that details some of the findings and recommendations that we presented today, if you're interested in learning more. And just finally, I'd really like to thank a big thank you to all of our partners, project partners, and funders, as without them, this work would not be possible.